does mental health really mean? As a few people have already been saying this phrase, it really means brain health. And what is your brain? It is billions of neurons that are wired together into complex circuits by literally trillions of connections, or also called synapses. And you might be able to think of your brain a little bit like the electronic schematic inside your iPhone, except that the biological circuits of your brain, illustrated here beautifully by a neuroscientist artist Greg Dunn, also have this sea of neuromodulators washing over those circuits and changing how they work. And that includes tiny amines and large peptides, and believe it or not, even gases dissolved in the fluids of the brain. And that's all called neuromodulation. And that is what our lab studies. Reasons to study neuromodulations are, first and foremost, is its enormous relevance to mental health. Any mental health, neurodevelopmental or neurodegenerative disorder you can think of, has some core feature of neuromodulation that is either perturbed in it or perhaps offers some therapeutic intervention potential, or likely both. Neuromodulation controls every essential behavior you might think of, and it really makes you who you are. I love that it literally often, sometimes figuratively, rewires your neural circuits throughout life. And because it's biologically fairly complex, my lab synergizes some tool development work together with biological applications. And that includes neat things such as using lasers to make new synapses grow, which I'll mention today, building new kinds of microscopes, and using very miniaturized tiny wireless tools to enable truly naturalistic behavior emerge in groups of animals. So this is the best place in the world. I feel I could be giving this talk right now. But it's also a very special time. In addition to it being the anniversary of 9/11, it's also the end of a National Suicide Prevention Week, right? This is the last day. And I'm not going to belabor all the key statistics. They are dire, and you have probably seen them, and they're especially bad for our younger population. And they are especially been exacerbated by COVID, along with every other aspect of mental health situation that you can imagine. And so we need some fixes, and the fixes that we currently have aren't quite enough. And I think one aspect that's really important here is to push advancing the rapidly acting antidepressant. That's especially critical if you're dealing with suicidal ideation. You just don't have time to go through the 6 weeks that it takes for conventional SSRIs, such as Prozac, to work. You just don't have time to be switching and trying alternative medication and trying to optimize the doses. We're losing people. And so treatments that work faster, treatments that work for more people. And we've got to start taking resilience seriously and study it the way that we study cognitive enhancement, the way that we try to increase longevity, because we can potentially make some difference and create means, pharmacological or otherwise, for enhancing our resilience in the face of stressor. And of course, extremely important is the work that's been so championed by OneMind, is destigmatizing talking about mental health. In the society, broadly speaking, but even in science, us scientists are supposed to be all data-driven and abstract and impersonal, and it kind of forgets the fact that we too, either ourselves or through our close family members, our loved ones, deal with mental health disorders directly. And so I want to extend a personal thank you to OneMind for empowering me, for example, to admit for the first time in a public setting that I've lost a sister to major depression and suicide. And so it would be a lie to say that that has nothing to do with my interest in resilience and fast-acting antidepressant, although in truth, it had preceded that said event. So what we really want to do here is, my pointer is not working, that's all right, is fix the human brain. It's working? Okay. No, it's not. It's all right. It's okay. It's fine. We want to fix the human brain. And sometimes we study the human brain in order to fix the human brain, and sometimes we don't. We study these furry creatures to which we're enormously grateful, mice. They may not be, monogamous pair bonders, but they sure have a lot of really complex behavior. And early in my career, I was really interested in how systems that involve dopamine reward modulators, so to speak, develop very quickly to enable really complex goal-directed actions. But over time, I've become interested in sort of the flip side, I would argue, as evolutionarily important form of learning, where we find ourselves in somewhat unpleasant situations or truly unpleasant situations. And that's the kind of space where resilience starts to matter. Because when we have two different people in the same unpleasant situation, their response and the consequences of that response for psychiatric risk probability are very different. So the work that gave rise to my Rising Star Award in honor of Nick LeDeit all starts with some projects that were recently published that were spearheaded by talented graduate students in the lab, Mingzheng Wu. And we really wanted to gain a better understanding of this really promising small molecule called ketamine that appears to be a very, very rapidly acting antidepressant working on the order of hours, and perhaps also transiently so. Biological insights into its action mechanism continue to be discussed and debated. We especially don't understand all features of the timing. And here, some prior rising stars have made enormous contributions. And Mingzheng and I realized that we think it's quite possible that dopamine, canonically associated with reward, but also important for processing aversive signal, I don't know where Stefan Lamel is over there, is potentially capable of explaining a lot about what ketamine does to both neuroplasticity and behavior. And so in order to do those studies, we used a very cool platform where we can leverage lasers to make new connections be born between neurons in an actual living section of the brain. So this is a neuron, and you can see its dendrite that collect information are densely studied in these tiny membrane protrusions, and each one of them holds an excitatory synapse for this and many other types of neurons. And so there are many, many connections per synapse. And we can use a laser to photoactivate some glutamate molecules right in that spot where you occasionally see this protrusion blossom. The movie is on a loop. And literally, at will, where and when we want, generate a new synaptic connection between this neuron. And this is a really direct causal way to assay plasticity, different from anything we have had before. And so this is that specific molecule. It's got a little tail, which we can clip off using a tightly focused beam of light. And then we can do this kind of assays, and some of the time new dendritic spines will be born. These spines start carrying a functional synapse within a matter of minutes. And we can prove that by literally recording the strength of individual synaptic current from one of these new guys and one of its older neighbors. And this gives us incredibly precise assay to interrogate the potential for plasticity on any neuron that we can target genetically with great specificity and as much spatiotemporal control as you want, sub-millisecond, sub-micron. So we did this a bunch of times, restricting green fluorescent protein expression, thank you, jellyfish, to a particular set of neurons in medial prefrontal cortex important for higher cognitive function where we knew ketamine is likely acting. And then we did a bunch of these inductions of the genesis of new spines, and we got some successes and some non-successes. And then you collapse it into a kind of a probability, propensity for plasticity measurement. And this is a great causal readout. It's compatible with any kind of pharmacological and genetic traction you might be able to do, and really nicely complements kind of in vivo long-term tracking in animals where you cannot do very rigorous manipulations. So using these kinds of assays, we discovered a bunch of things. First of all, a single injection of ketamine within a matter of hours dramatically enhances its potential for neuroplasticity. Secondly, having a bad experience tanks your neuroplasticity, but in a way that is recovered, again, by a single injection of ketamine. And this was the first time the light bulbs went off thinking resilience, is what we noticed using a simple assay where an animal could either fail to avoid a mild unpleasant stimulus or successfully avoid it. We realized that plasticity correlates with this resilience, meaning that when probability of spinogenesis, the potential for plasticity is high, the potential for failure is low. And so, ding, ding, resilience. So this finding of neuroplasticity and its extremely rapid regulation by ketamine helped kind of bridge together some effect timing differences previously observed in the field. So this potential for plasticity seems to work a lot faster than the time when people actually can pick up substantial structural changes in the brain that accrue over time. And to me, I think that means that what happens after, in the clinic, after a single dose of ketamine injection might really matter. This could be perhaps a time of significant therapeutic intervention, and that's not really currently something that's being done. So next, we wanted to try to delve a little deeper into the dopamine component of this, because I've told you, but I haven't really shown you any data suggesting it's dopamine dependent. Well, we show that pharmacologically, genetically, knocking out receptors, every different way it works. And just to give you a little bit of a sense of it, again, this uses the photometry technique, which has been very well introduced by every single of the junior awardees so far. But what we did with it is to track dopaminergic neuron activity. And we were actually created this metric of a trace distance, which means how much do your dopamine neurons differentiate when you fail to do something good for you and when you do it. And we found that this trace metric was very interesting because it predicted basically who is going to be not resilient and who is going to be helped versus not helped by ketamine. And that's basically biomarkers, right? And it's clinically significant because, first of all, we can measure aspects of dopaminergic function in humans. And secondly, because now that ketamine is FDA approved as an adjunctive for treatment-resistant depression, there are going to be people on dopamine-modulating drugs prescribed this thing. And I very strongly predict that this will totally determine the efficacy. So we realized we need to understand resilience better. And to do so, we have to get beyond those correlations and relationships and do more causal experiment. And my goal over the next 3 years is to answer 2 questions, very simple. First of all, does neuroplasticity potential actually predict behavioral resilience? And secondly, is it the mediator, is it required for when you try to enhance behavioral resilience by using things like changing dopamine signaling or ketamine effects? And so now we've learned, instead of making new synapses, we've learned to unmake them with light. And we want to bring these new tools together with this very selective deletion to combine it with our plasticity potential interrogation, measures of dopamine signaling, and behavioral resilience in assays that really maximize individual variability. And we'll be aided by a few other exciting projects in the lab, including using wireless tools to read and write brain patterns. If you're curious, the New York Times recently covered our work on related studies. And also, because our screen is, somewhat fast, but it's not that fast. We can't screen 100,000 drugs with it. We've developed a new, very, very large-scale screen specifically for neuroplasticity, and we hope to bring it to bear and find new drugs that help enhance resilience that we can then understand mechanistically and hopefully bring to the clinic. And with that, I'm out of time. I want to thank OneMind again. Thank you to Rabina, and thanks to my lab, and I'm done.